Okay, we're going to get started. Welcome everyone to Thought to Speech, a clinical trial for adults with cerebral palsy featuring Dr. Leigh Hochberg. This webinar is closed caption enabled and we've turned on auto transcription as an accessibility feature should you need it. I also want to let you know about the Q&A function on this webinar. You can type your questions for Dr. Hochberg by clicking on the Q&A icon and we'll get to as many as we can after the main presentation. I'm Jocelyn Cohen, Vice President of Communications and Engagement with Cerebral Palsy Alliance Research Foundation here in New York City. We're also known as CPARF, and it's exciting to have you join us for this exclusive event. Over the last 3 years, we have proudly provided funding to help move the Thought to Speech project to this point as it gets ready to move forward to human clinical trials. Funding this work to this point, learning about the intricacies, and understanding the breadth of possibilities has continually energized our whole team, showing us exactly why we do what we do, showing us exactly how life-changing cerebral palsy research can be, and how important it is to have people on our side to turn possibilities into reality. That's why we're here today, and we are truly grateful to have Dr. Leigh Hochberg with us. He is a leader of the BrainGate Consortium and the lead investigator on the Thought to Speech project. He currently holds several impressive roles, including serving as the director of the Center for Neurotechnology and Neurorecovery at Massachusetts General Hospital, professor of engineering at the School of Engineering and Carney Institute for Brain Science at Brown University, director of the Veterans Affairs RR&D Center for the Neurorestoration and Neurotechnology at Providence Veterans Affairs Medical Center, and senior lecturer on neurology at Harvard Medical School. He is deeply driven by BrainGate's guiding principle of turning thought into action, and that idea sits firmly at the center of the Thought to Speech project, which has the potential to revolutionize communication for nearly 4.5 million people with cerebral palsy worldwide. Please welcome Dr. Leigh Hochberg.
Thanks very much, Jocelyn. It is great to be here today, and thank you all for gathering to hear a little bit about our hopes for a Thought to Speech clinical trial for adults with cerebral palsy. I'm hoping to speak for somewhere in the realm of 30-35 minutes or so, and then I really look forward to hearing and answering your questions as best I can. I want to start off with a many ways most important slide, which is the great group of people who I get to work with every day. I'll start out. I'm not sure which corner of your screen he may be in, but I know that Sid Cash, who together with me co-directs the CNTR, the Center for Neurotechnology and Neurorecovery at Mass General, and Sid and I have been very much together in thinking about and planning for this clinical trial, going back 3 years to this week, which is when, as I'll show you in a timeline, a little bit later, the thought process, if you will, really began for this clinical trial. The group that I'm going to be telling you about, I've highlighted a few folks in green here. They're the ones who would be most intimately involved in the proposed clinical trial and research to help people with cerebral palsy, but they are part of an even larger universe of brain-computer interface researchers across multiple institutions. And I'm sure that you've seen many slides like this one before. And I do want to point out that, as is often the case, and is certainly true here, I'm going to show you some data, I'm going to show you some videos, and it's not the people whose faces you see who should really be credited for any of that. It's the people who are whose names are on the right side of the slide, who are our students, our colleagues, our researchers who have really kind of dug in to be able to think and to generate the type of research that I'm going to have a chance to tell you about. So I spend some of my time, and in fact, I was spending all of last week as a neurointensivist. That is, I work in the neurointensive care unit at Mass General, seeing people who have acute, often life-threatening diseases or injuries of the nervous system. And it's in bringing that experience and those interactions with our patients in the neuro ICU and our family and their families to the BrainGate research that really helps in many ways to generate some of our goals for the ongoing BrainGate research, and as well for this Thought to Speech proposed clinical trial. So what are some of those goals? So for somebody who has just had a spinal cord injury or a stroke, who I see in the neuro ICU, I want to be able to tell that person that they're going to be able to move again tomorrow. That's not something that I can tell them today. but it is something that I want to be able to tell them. And I think that the research that we're doing is going to get closer to actually being able to make that statement. For somebody who's had a brainstem stroke and has become locked in, that is awake and alert, can hear everything, can feel everything, but is unable to move or unable to speak, I want to be able to tell that person that they're going to be able to communicate easily again tomorrow, not next week or next month or next year, but really tomorrow. For somebody with ALS, I want to be able to tell somebody that they will never lose the ability to communicate despite this diagnosis of a progressive neurodegenerative disease that slowly can decrease mobility and decrease communication. And I mentioned spinal cord injury, brainstem stroke, and ALS because it is people with those diseases or injuries who have thus far been enrolled in the BrainGate clinical trials. But I know why we're here today, and it's very similarly that I'm hoping that these types of intracortical brain-computer interfaces, we will be able to say to people with cerebral palsy that you will gain the ability to communicate easily tomorrow. So for someone who is severely dysarthric or perhaps anarthric with limited mobility of their hands and restricted or limited communication, I want to be able to tell them that they're going to be able to communicate easily. And I'll show you in just a little bit what I mean more specifically by communication. So an overview, the topics I'll be going through, I'm going to tell you a little bit, perhaps an embarrassingly little bit, about brain-computer interface or BCI science and the types of signals that we can record in this particular flavor, if you will, of BCI research. I'll spend most of the time telling you about BrainGate for communication, reviewing some of the research that's been done over now the past 17 years. I'll then focus on what our goals of the TTS or thought-to-speech trial would be. And then we'll get into a little bit of the nitty-gritty about the clinical trial planning that's occurred to date, a proposed timeline for where we'd be able to go from here, and then, as I know is of some interest, what the financial summary is for being able to achieve these goals. So what is a brain-computer interface? I suspect that some, if not all of you, have had some exposure. Either you've heard about BCIs, sometimes called brain-machine interfaces, neural interface systems, whether it's the one I'm going to tell you about, which is in a BrainGate context, or whether it's any other BCI. All BCIs have three major components, which is a neural sensor, a decoder, and then either an assistive technology or a technology that can be controlled with the use of that neural sensor and decoder. So in thinking about a person and a neurologist, I've put a brain up there to represent a person. If that person has limited ability to communicate or limited ability to move, the first goal of the BCI is to be able to extract a neural signal that is relevant to the intention to either move or to speak. In order to do that, brain-computer interface, when we're kind of sitting at the desk and designing it, needs to be able to answer a few questions. We need to know what signals we want to record from the brain. There's lots of signals that we could record from, so we have to choose at least one or a couple of them that we think are going to have enough information to be useful. We have to choose an area or a few areas of the brain that we would record from, and we need to choose a sensor that would allow us to record the signals that we're interested in from the right areas. Then for any brain-computer interface, once we have that signal in our hand, so to speak, we put it into a black box. And that black box is the home of my colleagues in computational neuroscience, who in many ways have one of the most fun jobs in the world, which is to really try to understand the language of the nervous system, to understand what I've written here as being a firing rate vector, that is how fast or in what pattern is a particular neuron or groups of neurons firing in the brain. And if somebody's trying to move their hand to the right, we want to be able to re-enable that movement of their hand. And sitting between those two, sitting between the decoder, or sitting between the neural activity and the intended movement, is the capital letter A there, which is the decoder. All of the math, all of the computer science are reduced to one capital letter. It's actually where a lot of the research happens, which is filling in that letter A properly so that in real time, somebody can control an external device. Before leaving the desk, we have to think about what is that device? What is it that we're hoping to restore? What type of communication or mobility are we hoping these systems will provide? I've put a list there. I won't go through them in detail. We're going to focus as we go forward in thinking primarily about communication, that is restoring the ability either to type or providing the ability to type easily on a computer screen, or decoding speech, that is decoding the intended vocal verbal output and converting that in real time into audible speech, for example, one that would be provided by a speech generating device. Lots of other ways that these BCIs can be used, and I'll be happy to chat about those in the Q&A. So this is the only other science slide that I'm going to present, and it's just to give you a quick impression first of the types of signals that we're recording. So right now it's only the upper left-hand corner video that's playing, and what you see on the screen are a bunch of pink, blue, and yellow squiggles that seem to be flickering on and off. Each of those little squiggles represents the voltage, or that is the signal that's being recorded from a single neuron in the brain. And in this case, it happens to be the single neuron in the brain of somebody with advanced ALS, that is advanced Lou Gehrig's disease. So all of that neural activity, and I guess you'll have to believe me that that's a lot of neural activity that we're recording, because most of those boxes that are black there are filled in by colored squiggles. That is a lot of neural activity being recorded from the cortex of somebody who is unable to move, is unable to speak, and is mechanically ventilated. But that neural activity is modulating or changing as he attempts to move his hand. I'm going to press play in just a moment. You're going to be listening now to somebody who's speaking and asking one of our participants in the BrainGate trial, who's had a particular sensor placed in the brain that I'll tell you about, as she tries to do something she's unable to do, which is to open and close her hand.
Close your hand. Relax. Open your hand.
So as she tries to open her hand, which again is something she can't do, you see a lot of pink on the screen. You can hear what is music to my ears. That is the sound, if you will, of a neuron firing away. And that's just a single neuron. And if we think about just how informative that neuron is, when it's quiet, we know that she's not trying to open her hand. And when that neuron's making noise, that is, when it's firing, we know that she's trying to open her hand. If we multiply that by dozens or hundreds of neurons in the attempt to either move in a more complex way one's hand, or perhaps be able to speak, that's why this type of technology, I think, and the group that's been working on for so long, has the potential to restore or provide the ability for somebody with cerebral palsy to be able to communicate in an easier, faster way than they may be able to without this type of communication technology that's currently in development. All right, that was a very brief scientific overview. Happy to talk in much more detail, but let me jump ahead in some ways to some of the research that's been done using the BrainGate system, which is an investigational medical device. We're a group of academics. I will mention in a little bit some of the institutions that we currently work at, and that continues to do the research. But let me show you some of the publications that have come out of that, and some of the communication-focused research. So first, let's focus on what is the technology. So the technology is what is sometimes called a BrainGate array, or a Utah array, or a BlackRock array. A picture of it is in the kind of lower middle of the screen. It's a 4x4 millimeter array of electrodes that can be tapped into the top of the brain. I'm now going to do something that I'm pretty sure won't work at all on Zoom, but I'm going to hold up as best I can. I'll put my hand up to this tiny array that I'm holding up. This tiny array, about baby aspirin size. There's a picture of it against the head of a penny there. 4x4 millimeters, has 96 active electrodes on it. 100 electrodes total is listed there. And each of those is able to record from one or more neurons in cortex. It's connected by the fine wires that you saw to a percutaneous pedestal. That is a percutaneous pedestal that protrudes through the top of the head and through the skin. And that small pedestal can be connected to either a cable, as shown in the diagram here, or, as I'll show you in a little while, to a wireless transmitter. And if everything works the way that we want it to, somebody with tetraplegia, currently in the BrainGate trials with either brainstem stroke, cervical spinal cord injury, or ALS, would be able to control a cursor on a computer screen simply by thinking about the movement of their own hand. There have been 14 participants in the BrainGate clinical trials to date. That is roughly about half of the world's experienced in having one of these arrays chronically placed in the brain for neuroprosthetic research. Based on what we were able to start back in 2004, there are wonderfully now several other academic institutions, both in the US and around the world, that are using this array chronically to try to develop systems that will help people with paralysis to restore their mobility and also restore communication. Our consortium, which is just is both incredibly fun to work with, incredibly rewarding, and the hour prior to joining us all here today was our weekly BrainGate meeting, where there's sometimes 40, 45 folks from around the country who are gathered from these institutions, which are Brown, Case Western, Mass General, Stanford, and the VA, all working together. Incredibly close collaboration. And most importantly, what this allows us to do, no matter how complex the science itself may be, it allows one group of investigators on one coast to have a hypothesis, to have a new idea for how to decode neural activity, to work with their local clinical trial participant, and then to take that idea and send it across the country, if you will, to another group of investigators. I'd call them somewhat independent. They're certainly in a different location. They're working with a different participant, sometimes with a participant who has a different neurologic injury. And asking the question, does that innovation, does that approach to decoding neural activity work not just in one person, but in two, or sometimes 3 and 4? And that's one of, I think, the real opportunities that the BrainGate Consortium offers when we think about launching a new clinical trial, which is to be doing research not just efficiently, but really to make sure that it's repeatable and it has all the rigor and reproducibility that's so important to make sure that the science and the progress that we're making is robust. Adding to this consortium, specifically for the Thought to Speech project, is UCSF and the great work that Eddie Chang has been doing in speech decoding. Get a chance to chat with Eddie on a regular basis. He also is working closely with the BrainGate team at Stanford, and we've, as I'll show you in a little while, and some of the meetings we've had about Thought to Speech has been really integral in helping us to collectively think, how do we move this trial with this technology forward in a way that we can learn quickly and effectively from really multiple laboratories kind of background in this type of speech decoding. All right, so let me jump ahead. You are looking with the same perspective of one of the participants in our clinical trial. Happens to be somebody with ALS, somebody who is mechanically ventilated, and he's thinking about the movement of his own hand that he is unable to move. And as he thinks about moving his own hand, he's controlling that little white cursor on the screen. He's moving it to the target. And when he gets there, he's clicking by imagining or attempting squeezing his hand. I know that wasn't that exciting of a video. But what you saw was somebody who's unable to move their hand, thinking about moving their hand, getting to target on a screen fairly quickly, fairly smoothly, fairly accurately, and being able to click, that is, being able to point and click. The real reason that I'm showing you this, though, is to focus not on what's on the screen in the middle, but on the background, is that all of our research now for 17 years since the BrainGate trials were launched occurs in somebody's home. So as convenient as it would be, and there could be some advantages to developing a technology at the university laboratory or the medical center laboratory, which we can shield of electrical noise and we can optimize in every way possible. We've always wanted to make sure that we can develop and test this technology where it actually has to work, which is in somebody's home. And that's what we've been doing, and that's what we would propose to continue to do as part of the TTS trial. Move forward a little bit more. You're again looking at the screen of, here again, a user in the trial with ALS who's pointing and clicking on this somewhat unusual keyboard. The keyboard happens to be arranged in the order of frequency of the letters in the English alphabet, which is why THE is across the center. And she is copy spelling. She was asked to spell out that phrase that's at the top. And you can see the speed with which she's pointing and clicking quite accurately. So she gets to the letter of interest. She imagines or intends to squeeze her hand and types out that letter. This was a publication from back in 2017, getting upwards of 39 plus correct characters per minute. There's no word prediction being used here, which is a well well-used technology that could clearly be incorporated. And at that time, now 3.5, 4 years ago, this was about 10 times faster than the most rapid scalp-based brain-computer interface that was being used by people with paralysis. So when we as a BrainGate team saw this speed of control, and I'll pause this for a moment, when we saw this speed and accuracy of neural control of a cursor on a screen, we asked ourselves what probably seems like a somewhat obvious question, which is why is our group of neuroscientists and neurologists and neuroengineers developing user interfaces? There's actually plenty of good user interfaces that have been developed over time. And now you're looking at two people with ALS. I'll tell you more about what's on the top of the head in just a moment. Both mechanically ventilated. The woman on the left has one of the arrays placed into the brain, and therefore one of these cables that's protruding or connected to the top of the head. I'm going to make that cable go away in just a moment. In the lower right, gentleman also with ALS, two arrays in the brain, two of the cables that are connecting to the rest of the recording hardware. And they are both using completely unmodified tablet computers. So she, on the left, is sending a standard, these are Google Nexus tablets. She's sending a standard Google email to Paul Nurjukian, who's at the time, one of the postdocs now on the faculty at Stanford. And she's using that much in the same way she would have used that tablet computer prior to the onset of weakness of her hand due to ALS. In the lower right, he's on his tablet computer, has brought up YouTube. He's doing a search for ALS and to learn more about the disease that he has. So it's when I see these two types, and I'll pause these as well. When I see these two types of control, easy, intuitive control over a industry standard technology, that reminds me of what one of our original goals is to be able to restore communication for somebody who's lost the ability to communicate, or to provide communication rapidly for somebody for whom communication may always have been challenged. And this, using a tablet computer that we can buy off Amazon and have delivered in an Amazon Prime 48 hours later, those are the technologies that we really want to be able to provide access to, at least as one step of a thought-to-speech clinical trial. Not an assistive technology, which by their very nature have limitations, but a technology, a communication technology with all of the power and all of the flexibility that industry has brought to people who are able-bodied, all of that being brought to bear so that somebody with cerebral palsy or other forms of paralysis would be able to use that same device with the same ease and intuition as anybody else. So a question that often comes up is, okay, so you're recording from the brain, and somehow you got to fill in that black box that I was telling you about earlier. You need to build the math that decodes the brain activity. And the question is, how long does it take for somebody or the system to learn to decode that brain activity? So in a moment, I'm going to press play, and you're going to see what we call a center out task. Our participants, there's two of them, one on the big screen, one in the upper right. They're being asked to try to move their hand towards the target that's highlighted. And when I press play, the decoder, the black box, knows nothing. It is naive. It has no prior knowledge of what these brain signals are going to look like. And not surprisingly, when a person is trying to move their hand towards that target, the cursor is appropriately, because the decoder knows nothing, kind of flying off into space. So we reset the target to the center, and we again ask the participant, try to move your hand towards the target that we've highlighted. So what we know kind of on the software side is we know where the target is, and we know that the participant's always trying to move their hand towards that target. So informally, we can regress, if you will, the neural activity against the known intended direction of that person's hand. And now that about 40 seconds have elapsed, we went from a filter that knew nothing, that is a decoder that knew nothing, to what is actually not bad two-dimensional control of that cursor. And again, that cursor is being driven entirely by neural activity that's generated by the simple thought or the simple intention to move one's hand towards the target. Now, there are two of these on the screen. The upper right, which you may have been looking at, or perhaps not, basically the exact same video. But in that video, it nobly says participant T5 trial day 30. It was the very first time that he had ever used a brain-computer interface. So we had seen what you saw on the main screen there to begin with, which is that we could train up one of these decoders in what appears to be just tens of seconds, would continue to get better over 3 or 4 minutes. In this case, we did this again. We said, okay, go. And the people who were in the room watched, and the cursor did nothing for the first 10 seconds, and then 20 and 30 and 40. And then for the first minute and up to 2 minutes, the cursor was just floating around aimlessly. And it was at that point that the participant then turned and said, when would you like me to start? So the team, I guess, had a sigh of relief and said, well, hang on for just a second, hit reset, and said, okay, how about starting now? And again, even though it was his very first time ever using a brain-computer interface, within tens of seconds, there was the emergence of two-dimensional control of that cursor, which then continued to get better over 3 or 4 minutes. So in those earlier hopes for this type of technology, restoring communication and doing so quickly, meaning tomorrow, these types of machine learning, these types of approaches to decoding neural activity, I think is going to be really powerful towards providing rapid control over external devices. Everything that I've mentioned so far is about control over one's hand, for control over a tablet computer, for example. We've been recording so far in the clinical trials from a part of the brain that is involved in control of the hand. But even though we've been in a so-called hand area of the brain, we've also asked our participants to speak. And what you're looking at here is neural activity, as one of our participants is speaking a few different phonemes. And you may be able to tell that each of these little dots happens to be a neuron that's firing as these various phonemes are read. And it looks like each of these different phonemes has a slightly different pattern that's evoked in the neural activity. And without going into detail on the matrix on the right, really what that's showing is that we're able to decode with about 84% accuracy which of 10 phonemes somebody is speaking from just the neural activity in the wrong part of the brain. We're recording here from a part of the brain that's really involved in controlling the hand. So when we saw that we're able to extract relevant neural data relevant to speech a couple centimeters, if not more, away from where we know speech is primarily encoded in the brain, that gives us great optimism for a thought-to-speech trial and for being able to use these types of technologies when we have the opportunity to record from the right part of the brain to be able to extract intended speech and to decode it in real time. You may have seen just in the past week, one of our BrainGate team's most recent publications about decoding handwriting. I was showing you point and click before. Here, what you're looking at, these squiggles on the screen are what we would call the neural trajectories. They are the neural drawing of these letters. So one of our participants with spinal cord injury, unable to move his own hand, is drawing or attempting to draw each of these letters. This is the neural decoding. This is what the letters look like if the neural activity itself was actually drawing the letters. And what that enables is an even more rapid method for communication. So here, and this was just published in Nature last week, here's our participant. Again, there's a big thing sticking out from the top of the head that's attached to the small pedestal. I'll tell you about that. He was asked an open-ended question, where were you born? And we're watching in real time his attempted handwriting and being decoded into the text that you see on the screen. And this is also without the benefit of what we can add to it, which is a little bit of neural data up there on the right, an even additional layer of increasing the accuracy of this. It was already about 94% accurate for the letters to begin with. And if we add a simple language model to that, we can get upwards of 99% accurate handwriting decoding at this speed. So I'm really optimistic about being able to use, for example, attempted handwriting as one method for communication, either for brain to text or brain to computer generated speech. there's been this big cable that I keep showing you. I keep telling you that we're going to make it go away. We have made that big cable go away. We can now use what I hope you're able to see on the screen is I'm holding up this wireless transmitter. It is now this device that can be placed on the percutaneous pedestals. It wirelessly transmits the full bandwidth of neural data to nearby antennas that allows for our participants to be able to use the BrainGate system at home with the assistance of a caregiver, and with us either not being present or us dialing in remotely to help to set things up and then for them to be able to use it subsequently. And just to show you one video of that in action, this is one of our participants. He's laying on his right side. You may be able to see one of these kind of blurry wireless transmitters. And I'll press play. I'll speak a little bit more loudly. This is just Pandora. And he's thinking about using his own hand. He's controlling the unmodified little cursor on the screen.
Okay, yes. Give it the thumb. There you go.
And even though that's a very small icon, much like somebody who's able-bodied went over to it, liked that music. And in many ways, the goal here is just to show that we're not trying to develop an assistive technology. We're trying to enable control of standard technologies with all of their power. In this case, done so wirelessly. And again, you can probably tell in his own home. Last thing that I'll show you, just to speak a little bit more to the breadth of the BrainGate research. We're largely focused on restoring communication. We also want to be able to restore mobility, do the research that will allow people to have restored mobility. I'll hit pause here for just a moment. This is a gentleman with a cervical spinal cord injury. You can replace, in your own minds, these two big cables with wireless transmitters. In addition to the neural decoding that we're doing, there are also pairs of electrodes that have been placed in the muscles about his elbow and hand. He does not, on his own, have the ability to flex and extend his elbow or to open and close his hand. But here he's thinking about bringing the food that he requested, which are these mashed potatoes, to his mouth. And as he thinks about doing so, right now he's thinking about lowering his arm. That's actuated by the mobile arm support that he's under neural control of, but because there aren't muscles about the shoulder implanted, that's a mobile arm support that's doing that. But his holding of the fork, his ability to flex and extend his elbow and to bring those mashed potatoes to his mouth, that is rudimentary. It's simple in that the movements themselves are far from dexterous or fast, but it's early steps to being able to reconnect brain to limb and to restore here upper extremity mobility for him to be able to, for the first time in years, to be able to feed himself solely of his own volition.
435.
Thank you. That was Sid telling me that I should keep going. We're working towards fully implanted systems. And with that, I said I was going to spend most of the time on communication. Let me get through a few goals. So what are our trial goals? In the thought-to-speech trial for adults with cerebral palsy, we want to enroll 2 people per year for 3 years. There will be clinical trial sites at MGH and Stanford. Brown and UCSF will be involved as scientific sites. We have collaborators across multiple institutions, both in the US and elsewhere around the world. And as mentioned, we're hoping that each participant in the trial would be enrolled for at least 2 years. Goals include demonstrating intuitive, rapid, text-based, and as you saw, speech-based communication at home. And I don't want to understate inadvertently the neuroscientific challenge and excitement and importance of the fact that this will be the first ever insight that would be gained into neural decoding from cortex in people with cerebral palsy. All of the videos that I showed you thus far are from people who had lost the ability to have dexterous movement or fluent communication. And it's going to be a really important, exciting, and I think very possible step to be able to decode this type of neural activity associated with the intention to move or speak with the participation of people with cerebral palsy. From the beginning, we've been thinking as well about children. This clinical trial will be started appropriately with adults who join us, but we want to make sure that we're learning as much as we can so that these types of technologies would be able to be brought to children as well. The devices that we'll be using look somewhat like the ones that are on your screen. There would be 2 pedestals protruding from the top of the head, allowing us to record from 4 arrays from 4 different sites in the brain, again, all wirelessly enabled. Moving to the clinical trial planning. I want to make sure that I leave time for questions, so I'll move somewhat quickly. Main point is the one at the top, is that our clinical scientific regulatory groundwork is complete. And it's complete as a result of 3 years, which I won't go through in any detail, of meetings, of collaborative, both in person and more recently on Zoom, beginning with the CPARF Assistive Technology Summit that was held in San Francisco 3 years ago this month. Very rapid proposal to think about using BrainGate for a thought-to-speech system. Thanks to CPARF's incredible support for clinical trial planning over the past couple of years, we've been refining the clinical trial plan. And importantly, last September, we received both FDA and IRB approval for this trial for adults with cerebral palsy, including the ability to record from speech-related areas of cortex. I know often when I hear about early ideas of a clinical trial, there's a lot of this groundwork left to be done. We are now at this point with approvals in hand that would allow us to start much more quickly than if we were much earlier on in this process. Getting to a timeline, where would we be? If funding were to become available to do this research, we would celebrate, and then we would get back to work. It would probably take about 2 months to finalize the IRB protocols and obtain kind of the last iterative approvals between the institutions to know exactly, guided in part by CPRF, of exactly how we want to recruit and from whom. About 2 months later, we would then submit the final FDA supplement. They've already approved the trial, but we would be finalizing informed consent forms, getting the investigators certified. Generally would take a month or 2, probably at most, for FDA to provide their final approval. Could be faster than that because we already have the initial approval. We would then launch the trial, which is the first time that we're allowed to be advertising publicly for it and recruiting participants, though we can be speaking informally about it months beforehand. We then recruit our first participants, 1 each in Boston, Palo Alto. The device would then be placed probably about a month or 6 weeks later. And within about 3 weeks of that, we would begin the initial testing and home use of a BrainGate thought-to-speech system with the first 2 adult participants with cerebral palsy. So that's the timeline. It could be shorter than that. I'd be very surprised if it was longer because of all the groundwork that we've done so far. And lastly, I was asked to mention just a very broad financial summary of what this type of trial requires. So our approach to doing this is, as mentioned, we've been doing BrainGate research for going back to 2004. BrainGate research was started by a small startup company, Cyberkinetics, that had invested $10 to $15 million in investor capital itself. That didn't cover any of the cost of the university-based science. Over the first few years, there was additional NIH contract support that provided for some of academia's engagement in the original industry-sponsored clinical trial. Since 2009, all of the research has been funded by either the NIH, the Department of Veterans Affairs, institutional funds at the institutions listed there, more recently HHMI, that is the Howard Hughes Medical Institute, and from philanthropy, including philanthropy from and the support from CPARF. Numbers coming up in a moment, but I can assure you that we've budgeted for success, and we did not try to squeeze this proposed trial into a budget that doesn't achieve the goal. I think most people who are doing NIH funded research are quite familiar with severely under budgeting for their proposed outcomes. We did not do so here. The goal is really to be able to make sure that we have every opportunity to achieve our goals in a clinical trial and not trying to undersell it in any way. We also plan to purchase some of the equipment in year 1, which happens to be expensive. And the reason that I think about philanthropy being the key here is that the study can start sooner. It can proceed more efficiently. There's much more control and input from funding agencies, from philanthropy in this case, and it helps that much more to have vital community engagement. Finally, as mentioned, these types of preliminary data that can be achieved as a result of philanthropic funding are really, and by preliminary data, I mean the experience of working with the first few participants in the study. That's what allows us to then write a grant to secure federal funding to begin the research and continue the research with adults and potentially begin research with children. So the leverage there, it can really be substantial. And I should just mention that we may have budgeted for success, but my goal at every moment is to actually reduce the costs of the trial by reducing the costs of equipment and finding efficiencies wherever we can. It's expensive because most basic research discovers, clinical research tests, but here we're going to be testing and discovering and testing and discovering every week. The equipment happens to be expensive. That's equipment that we buy from a company for neural recording and for recording brain signals. That cost will come down over time. There's a neurosurgical event, which is an inpatient, so that contributes to costs. There are full-time neuroscientists and technicians and part-time clinical researchers that need to be supported in order for this trial to be done both safely and to collect all the data that are needed, and to convince the scientific community it's important that we're publishing papers along the way. I think this is my second to last slide. There's a wide range of potential budgets that could be applied to this trial. A 6-participant trial, which, just to go through this, a 5-year trial that has 3 participants per site, that is 3 on the East Coast, 3 on the West Coast, with a certain amount of other equipment that I can talk in more detail about, would have a total cost of about $12.7 million. If we wanted to recruit 1 participant each on the East Coast and the West Coast for a trial that lasted about 2.5 years, that total cost would be somewhere closer to $5.1 million. I can go into more detail on that if of interest. So with that, I just want to, again, highlight the great team that's engaged in the BrainGate research, to thank CPARF, thank Chris and Jean-Louis and Jocelyn and everybody at CPARF who I've had the chance to interact with together with Sid over the past few years. And thank you for your interest in hearing about this potential clinical trial. And I will pause there and be happy to answer any questions. Thanks.
Thank you so much, Leigh. I've been involved with CPARF for over 2 years, and every time I speak with you, I learn something new. And I hope that everyone that attended today learned something. And we do have a question and a little bit of feedback, and it says, "This is fascinating and thank you. Will there come a day when the computer will be able to pick up the intended hand movement without any direct connection to the cortex?"
It's a great question. And Jocelyn, if you can, oh, there we go. I'm going to unshare my screen. There we go. That's a little better. It's really an important question. And let me start with one statement, which is that if we thought that we could achieve these goals of rapid, powerful, flexible, intuitive, reliable neural control over an external device without requiring surgery, we would absolutely do it. There is no reason to do a surgery if it will achieve the same outcomes as any research or any medical device, for that matter, that doesn't require the same surgical intervention. There's some risk that's involved in any surgery, so those risks and benefits are constantly on our mind. I do hope that in the future, there's a way to pick up these neural signals from a distance, and from a distance actually would mean from the top of the head. But the physics of this remains that the very tiny voltages that are generated by neurons that sometimes are 50 microns, a really big neurons, 50 microns in diameter, in order to listen in on the neural activity of that fundamental unit of communication in the nervous system, which is a neuron, kind of need to be at the moment about 50 or 100 microns away from it. Once you get too much further away from it, you can no longer know what that one neuron is saying. And currently, if recording from the top of the brain or even the top of the head, you get a much more spatially smeared signal that, so far at least, has much less information in it than can be extracted by using a surgically placed device. So I share the hope that in the future, there may be a way to get these signals without requiring surgery. But right now, we think that the best way to have reliable, flexible control over an external device and to be able to decode speech with a really limitless, a full vocabulary without restricting the vocabulary to just a few words, that's probably going to require, at least for now, recording from inside the brain.
Understandable. Thank you for that. The next question has several parts to it. Are people considering marketing this to the mass market? If not, why not? Marketing to the mass market would bring more research funding and enable people with disabilities to become aware of it.
So when I think of this, when Sid thinks of this, the market is really in the context today of restoring or providing for communication or mobility that is currently impaired. So there's a market for these types of technologies that I think are clearly in the realm of rehabilitation, restoration, providing abilities in a way that they're currently not available to individuals with neurologic injury or disease. That market, I think, is actually substantial because it's more than what we've been discussing so far today. There's actually a much larger market of something that's called closed loop neuromodulation for people with, for example, neuropsychiatric disease, other movement disorders, and or seizure. One of the most important kind of future opportunities for providing personalized neurotechnology therapies is to record high resolution signals, to know what those signals mean from the brain, to decode them in real time, and to return a useful signal back to the nervous system. So the numbers of people, if you're to the question about a market that could potentially be aided in some way by this type of technology, I think is much greater than might initially be considered. If the question was about marketing to people without a neurologic disease or injury, I know that those discussions happen. I am totally focused on the potential rehabilitation and neurorestorative uses of these technologies. They may have some other uses as well that may bring in funding. And there are companies, regardless of what they're focused on. One of the really nice things about this field is that, in part as a result of the BrainGate research that's been going on for the past 17 years, there are now a number of startup companies that are focused on developing ever better recording devices for any number of medical or potentially other purposes. And I think that's a good thing for developing systems that will help people with paralysis.
Building off of that, someone asked, is Neuralink, Elon Musk's company, doing something different from what you and your colleagues are doing?
So important upfront disclosure, both Sid and I at MGH, that is at Mass General, have a clinical research support agreement with Neuralink. So we do have the opportunities to interact with the folks at Neuralink on a regular basis. I'll only, of course, share what is publicly shareable, and what they've said in the past. As you may know, Neuralink is developing a different type of recording electrode. That is an electrode that can be placed in the brain in a different manner than the Utah array that we're currently placing, and that records and is fully implanted. And all of those things, I think, are good. And when I just referred to companies that are developing systems that are going to move this field forward, I am very hopeful that all of those companies are successful and that they're able to develop products that will then be available to help people with neurologic injury or disease. It is a different approach to doing it. We will collectively find out when those clinical trials start how well they work for people with one form of paralysis or another. And I hope, as many people do, that they and others are successful.
Leigh, can I add in a little bit there, if it's okay? I think it's important to emphasize that people like Neuralink and other companies are developing alternate technologies for obtaining the kinds of data that Leigh have been talking about, single unit data. But the fundamental questions of what do you do when you have that data? How do you use that to understand what the brain is trying to do are relatively universal. So a lot of what's been done in the BrainGate clinical trials and that we're trying to do here in the trial that we're talking about would still be applicable to other technologies that are being advanced. And those just might offer, higher fidelity or higher ease of use or things of that nature. But the fundamentals, the basic neuroscience that we're looking at here will still be applicable there and still be very useful, regardless of the ultimate technology that's found to be, best for whatever reason.
Thanks, Sid. And I'll just add, the reason that Sid and I are engaging with the companies is because we want to do anything we can and everything we can to help them think about the deployment of these technologies. We want to make sure that they're designed to be successful in clinical trials, that the clinical trials themselves are designed in a way to maximize the information that's learned. And so anything that we can do to help them to be successful is, it's very much part of our mission.
Thank you for that. The next question is, aren't there many possible scenarios for why a person affected by CP is nonverbal? And does this mean you need more participants to have a broader sample and more robust conclusions?
Great question. Thank you. I'll start with what I continue to find to be a really interesting observation across the BrainGate trials to date. So there have been people in the BrainGate trials who have had spinal cord injury, brainstem stroke, or ALS, 3 different diseases or injuries. There are people who have been from about 24 years old to 65 plus. And the time between their injury or disease onset and when we placed the arrays in the brain have ranged over many years. So no matter how many axes we put those on, those 14 people that have been in the BrainGate trials and the other 14 or 16 that have been in other trials are all really unique individuals with a very, where one would think that the approach to decoding may be different, despite the fact that we're always, of course, recording from the brain, but recording from people whose nervous systems have been injured at various times in various ways. And despite that, we have been able to use the same constantly evolving neuroengineering, neurostatistical approaches to decoding brain activity with continued success and continued growth in accuracy and speed and reliability of decoding those neural signals. And I think, and I hope that the same thing is going to be true as we begin to learn from people who join us in a clinical trial with cerebral palsy. So absolutely, people with cerebral palsy have different injuries in different parts of the brain. I think that the methods that we're currently using to decode that brain activity should be robust to those differences. And while every individual is, of course, individual and each person's neural activity is unique, the mathematical approaches that are deployed in order to decode that neural activity towards decoding speech or decoding intended hand movement, I think are going to be similar enough that we're going to be able to make progress reliably.
That's great. So the next question I have, and a bit of comments as well, dovetails with the last question. And they say, thanks, Leigh, this is absolutely incredible. In the budget, there are 2 scenarios, 6 people at $12 million over 5 years, or 2 people at around $5 million over 2 years. Having more people to learn from us obviously better, and more time will offer more, I'd have to assume. Or could the 2-person trial with the shorter period offer knowledge that could bring this to millions of people?
So there's a lot to talk about there. It's a really important question. So the goal at the end, not necessarily of the first clinical trial, but I think everybody's goal is to have a system that is proven to provide improved communication or improved mobility for people with cerebral palsy. And that, when we collectively as a community are at that point, that's going to be a system that's manufactured by a company, that is supported by a company, that a physician can prescribe, that a surgeon in this case can place, and that the recipient, that is the user, can have confidence is going to provide for reliable either communication or mobility or both. In order to get there, we need to learn as much as we can, as quickly as we can, with the extraordinary participants who would join us in these initial clinical trials. I think neither 2 nor 5 would be enough of an experience to get to the point of saying there is now a product, if you will, that is ready to be launched. But even the first 2 to 5 people who are in the trial provide for iterative learning, continued evolution of the neural decoding approaches, learning all the things that may be unique or different about recording from the cortex of somebody with cerebral palsy, and using that information in a useful way for control of an external device. So the 2-person example is, the reason I put that there is that it is the clear minimum preliminary data that, for example, would allow Sid and I to then write a grant to the NIH that would propose that we could continue doing this research in a larger clinical trial. So that's where the 2 came from. The 5 is the 5 or 6 people, 5 years, 6 participants, is there would be a lot that is learned there. And with those 6 participants, I think we would very much then be at the point of moving next to what could be a pivotal trial. That is the trial of a device that's no longer constantly being iterated, but a system that could actually be tested for efficacy towards the point of getting some type of market approval. So that's where that kind of 2 and 6 come from in terms of participants, or 2.5 and 5 years come from. And there's lots of different ways to think through that in detail.
Thank you for that. We're right at 5 o'clock, basically. There were 2 questions that we couldn't get to. I'm going to go ahead and send those to you after this, and hopefully you can get some answers to me, and I'll get them to the person who asked. So just in closing, I want to thank you again, you and Sid, for sharing all this information with us. We appreciate you taking the time out of your afternoon to tell us about this transformative work and the ways people can help us move it forward. And thank you to all of our attendees for spending the time with us, for being open to learning about thought-to-speech technology, and for potentially helping us change what's possible for communication and cerebral palsy. Hope you all have a great rest of your day, and thank you again.
Thank you, everyone. Thanks so much for this opportunity.
We appreciate being able to tell everybody what we're thinking.
We appreciate you.
Thanks. Thank you.